Okay, now we're coming to the home stretch. Almost done here, week 9, and once again, we're continuing our conversation on molecular biology. But now we're getting into gene expression. What do I mean by gene expression? Well, it's exactly what I've been hinting at the whole time. Once again, I'm going to my little triangle here, and we're going to see that those genes, those traits that are carried on that DNA in the nucleus of your cells, what do they do? What are they there for? Well, they code proteins. And remember, proteins are those amazing molecules that are the workhorses in your cell, that create those amazing structures and those enzymes in the cell. So we're going to see how does the DNA on these genes actually encode proteins. That's what gene expression is all about. In order to do that, we're going to go back to DNA and RNA structure and function. Once again, we already covered this way back in week 3, I think it was, module 3, when we looked at basic biochemistry and we looked at the basic structure of your nucleic acids, your DNA and RNA. But once again, we're going to go closer at the structure of that double helix of the DNA molecule. We're going to once again look at DNA replication, which coincides to the last module we talked about with mitosis, what happens when DNA replicates. And once again, we want to be able to distinguish between the structure of the DNA and the RNA, and how DNA in the nucleus will once again encode just the right RNA in order to code a protein. So there's various forms of RNA, and you're going to have to understand all the various roles of RNA within the cell. After we review that basic biochemical structure of those nucleic acids, then we're actually getting into gene expression. And once again, this is part I think is fun. This is interesting. How we're actually, once again, just going to make a copy, just a little snippet of the DNA, which will then synthesize RNA inside the nucleus. It will leave the nucleus, it'll attach to a ribosome, and then the ribosome will bring in the appropriate amino acids to create the appropriate polypeptide, the appropriate protein. So you should be able to summarize this process of what's called transcribing, transcription, of RNA, messenger RNA from DNA, and then translating that RNA, that messenger RNA, to protein. So it's transcription and translation. So you should be able to understand the whole basic process, be able to describe it, and in how the DNA, once again, encodes the proteins. Now, along with that, we're going to talk about DNA technology. So we're going to talk about the importance of DNA sequencing to the study of biology, to really understanding how life exists, if you will. It all goes back to the DNA. You want to be able to state the purpose of the polymerase chain reaction in DNA cloning and summarize some of the products that have been produced using various biotechnologies. Lastly, we're going to talk about genomics. We're going to talk about the Human Genome Project and summarize some of the goals of that Human Genome Project to actually map out the entire human genome, which is just a massive feat in human history. We want to be able to distinguish between functional and comparative genomics, as well as genomics and proteomics. So once again, within molecular biology, a lot to cover. We're coming to our peak now in our class. So let's go have some fun.